Welcome back, everybody. We are ready to start talking about how DNA replicates. So, we've talked about genetics and the inheritance of that DNA, and we've talked about mitosis and meiosis, where that DNA needs to replicate so that it can get divided into those two cells. But how exactly does it replicate, right? We haven't talked about those details yet. So, that is what we are going to be discussing in chapter 9. So, both DNA replication, when the entire strand is copied, that entire chromosome. And then we're also going to talk about copying just little portions of it when you just need one gene copied for gene expression, right? When you need to make adrenaline, your body doesn't copy the entire chromosome. It just copies that little bit that codes for adrenaline, and it makes adrenaline from that. So how does that happen? So that's what we're going to be discussing in chapter 9. So get your coffee. I've got mine right here. Get your paper and notebooks and pens, and we will get started. Let me get this chapter open for you guys. Okay, I like to start with a picture here, if it opens up, of Rosalind Franklin, who you've read a little bit about so far. And lost my little pen here. And this photo 51 that she this is this photo that she took. Why is this? I'm having trouble drawing on here, so just give me a moment to figure out why is this not working. Here we go. Okay, so this is the photo that she took that really, I'm not an X-ray crystallographer, but you can kind of see that this might give the impression of a double helix, right? That it's kind of two strands of DNA here. So this was really the eureka moment when she took this picture. They showed that DNA is a double helix. We're going to be reading about that, more about that discovery. So as you know, it's so important to understand the structure of DNA. And, before that was understood, there was so much that we couldn't do. But now understanding DNA, its structure and how it replicates, how DNA is then used to control genes and gene expression can really help us understand the basis of health, right? Does somebody have a genetic predisposition to a certain disease? And ways that we could possibly have gene therapy to fix that. It's also understanding DNA and RNA has helped develop these new messenger RNA vaccines. And we're going to be talking about messenger RNA. So to give you an understanding of exactly what is this that's being made in these mRNA vaccines and why does that work? Why does that make the spike protein? We're going to talk about this will eventually lead into the genetic basis of behavior so we can understand how behaviors are genetically based as well. And we'll have more talks on behavior later in the semester. And then also, as all of you probably know, DNA can be used for forensic studies and not just for solving crimes when a human leaves DNA at a scene, right? But it can be solved for conservation purposes. So either crimes where someone has illegally poached animals, or it can be used just to track populations and estimate population size for endangered species. So the things that we are going to be discussing, we're going to talk about the history of DNA, that discovery. We're not going to go too deeply into that because that's so many of our readings and I don't want to give too much away. We'll talk about DNA replication. So this is copying the entire strand. Gotta get my pencil working again. Blackboard seems to have changed everything. So this is the entire strand. And then we're going to talk about transcription, translation, and gene regulation. So this is copying just a gene. So just what the cell needs at that time rather than copying the entire strand of DNA. And then we will also talk about genetic damage and mutation. So what happens when DNA isn't replicated properly? What are the results of that? So here we've got a picture of these major players that you've started reading about. Watson and Crick were at one college. I think they were at Cambridge. And then Wilkins and Franklin were both at King's College in London. So they were, working in different labs, presumably focusing on different parts of this question and this mystery of the structure of DNA. And this is Watson here, and this guy is Crick. Okay, so the structure of DNA. You guys know a little bit about this. We talked about this in chapter 2, but we're going to go deeper into it now. So as you know, DNA is nucleic acid. So that means it's made up of these little bases called nucleotides, right? So these string of nucleotides make up a nucleic acid, and these could be A's and T's and G's and C's, right? But it's not just this little base, this A or this T, or the A's and T's are made up of more than just that little base. There's this nitrogen-containing base, but then there's also a sugar and a phosphate group, and those are on the backbone of the DNA. And they are, if we think of it as a ladder, I'll show you a picture here. This blue here, this light blue, is this sugar phosphate backbone. And then these rungs are nucleotides. And as you know, the A always pairs with T, and G always pairs with C. So as we're talking, there's a fair amount of terminology, and I just want to make sure that everybody understands all the terms that we're using so that it is clear as we are discussing all of these processes. So the genome, when we're talking about an organism genome, that's its full set of DNA. So for humans, that would be 46 chromosomes total, right? We have 23 pairs. So you have two chromosome 1s, two chromosomes 2s, two chromosomes 3s. All of those together are your genome. You have your genome inside every single cell, except for eggs and sperm, those have half of your genome, right? But in general, most cells in your body, somatic cells, have your whole genome in them. So you could take out any one cell and sequence your genome, and it should be the same in every single cell in your body. A chromosome is just part of that genome, right? So if we've got 46 chromosomes, one chromosome is one linear piece of DNA, right? That there. A locus is just a position on that piece of DNA. So if we think of a chromosome as a long street, a locus is just where exactly on that street you are. So, right, you could be at any one of these spots, and somebody could say, I'm looking at a gene that's at, locus 23, and then everybody could go there and be like, okay, we're going to count down, 23 bases, and we all know exactly where you are on this gene, and everybody could look at the same spot. So it's just giving an address. Gene on that chromosome. A gene is a sequence of bases that codes for a protein. So a gene's at a specific locus. You could say this gene goes from, position 23 to position 462, right? And that's how many base pairs it takes up. But a gene is coding for something in particular. So you have a gene for whether your knuckles are hairy, and you have a gene for whether you can taste NutraSweet as sweet or as something bitter, right? And you have a gene for making adrenaline, and you have a gene for making cholesterol. You have a gene for making hemoglobin. All of these things are just a sequence of bases that can be read to then make that protein. So an allele is then an alternate version of the same gene. So in the last chapter, when we're talking about inheritance and dominance, if we're talking about the gene for melanin, right, big R might be making melanin, and little r would be the recessive, that you're not able to make melanin, right? So these are the same gene. It's that same sequence of bases, but there's different versions of them, right? Or when we were talking about blood types, there are 3 alleles, right? There's the A allele, the B allele, and the O allele. So any one individual can only have 2 alleles because we only have 2 copies of every chromosome, but there can be more than 2 alleles in the population as a whole. So as long as you understand these terms, as I'm talking for the rest of this lecture, things should be making sense. Okay, so we can look up here a little more closely at this structure of DNA. Here are these bases on the right. So you've got cytosine, which is C, right? Thymine, T, guanine, and adenine. A, T, G, and C. And just look, and what do you see? So the ones on top, cytosine and thymine, are pyrimidines, and the one on bottom are guanine and adenine. Those are your purines. There's some obvious similarities between the two pyrimidines and between the two purines. I'll give you a second. And I'm sure you see that the pyrimidines are each one ring, right? And the purines are two rings. And before they'd figured out the structure of DNA, they knew that it was made up of these four bases, but they didn't know exactly how, right? They didn't know if it was one strand or two strands or three strands was a theory. And there was a scientist named Chargaff who did an analysis and found out that the number of C's is always equal to the number of G's, and the number of A's is always equal to the number of T's. And they didn't know the significance of this at the time, but this was one of his findings when he then analyzed one little portion of DNA. And then he analyzed a different portion, and they always came out, it could be 20% C's and 20% G's, or it could be 33% C's and 33% G's, but they always matched each other. And then this was actually a big clue into the structure of DNA. So once, Watson and Crick and Franklin and Wilkins, once they had theories on the structure, this was a really big piece of evidence that they were correct. So think about that for a second. Why would that be? Just the fact that the proportions of them are the same. And the reason is because now we know that C is always paired with G. So if you have 20% C's, you're going to have that same number of G's, right? And same with A's and T's. And we can also look, and you'll see that if C is always paired with G, there's always three rings total, right? If you've got a C that has one ring and a G that has two, and A is paired with T, there's always three rings across. So this makes it's nice and clean. Because Blackboard seems to have changed my little drawing ability here, so I have to go in and erase stuff. So now, if there's three across, before they knew that it was even a double helix, but let's say they knew it was double, but they didn't know that A was always paired with T. What if A was paired with A? Okay, that would mean you'd have four rings across for A. Excuse my drawing, right? And then let's say the next one was a T, and that would only be two rings across, and it would be like this, right? And then you had a C, which was also two rings across, and then you had a G that would be four rings across. Well, now we have this really irregular, weird structure to our DNA. But instead, by making it always three across, it doesn't matter if it's an A and a T or a G and a C, it's always the same distance across. And then that twists around on itself, but it's a uniform shape. Now, another thing that you'll notice, and I can remove some of my drawings here for you. If we look at this picture on the left, this shows how everything is put together. And so we've got our base that we've been talking about, the bases, and then we have the sugar and the phosphate. When we're looking at DNA replication in a little bit, you'll see it always says that there's a 5' end of a DNA strand, and then it goes the 3'. And since it's a double helix, right, it's two strands, the other one always runs in the opposite direction. Okay. And what they're talking about here is just the way the sugars and the phosphates are arranged. So the numbers of the carbons on the sugar are numbered. So this is actually 5. Attached to that 5 end, and then down here is 3. So this is the 3 end. So if the sugars are all pointing up in this direction, this is the 5' end, which means that if we looked at it from this direction, right, they're not attached to this part of the sugar. So it's just which way that the phosphate's attached to the sugar that defines the 3' and the 5' end. You guys do not need to memorize that, but it's just that's how it's determined. So as you can imagine, if there were another, if this was a whole strand of DNA and another base attached down here, you'd have your sugar and the phosphate group would be coming up this way. But the strand, so this is our phosphate group. The strand running in the other direction, your sugar would be here and your phosphate group would be coming off to the bottom. It would be going in the other way. So we've looked at mitosis and how in interphase, the DNA is replicated, and then in prophase, it kind of condenses, right? That DNA condenses, you can see it. How does it condense, and how does it then, get organized enough for replication? Well, if we think of DNA as like a really long piece of toilet paper, right? It's the super long strand. If you've ever had a puppy who's gone into the bathroom and like unspooled an entire strand of toilet paper, it can like fill the whole bathroom, right? And it's a total mess and it's all tangled up. That's how your DNA would be if it weren't organized. And it would be really hard to like make a nice neat copy of it if it's all in a big tangle. So the DNA actually is like condensed in a really organized fashion. So here's your double helix. That is what we always, what DNA looks like. But then it gets wrapped around these proteins, which are called histones. And this is basically like taking that strand of DNA and wrapping it around that cardboard tube that the toilet paper's on, okay? It just gets it all neat. And then it's like if you had multiple rolls of toilet paper all attached to each other, right? Because if your piece of toilet paper was so big that you couldn't put it around one spool, you'd spool around like some of it around one and some of it around another and some of it around another, right? So this is just that all of those histones kind of in a chain, okay? And then it just can get further condensed and kind of squished together. So here this is what is happening as that DNA is condensing. It's always wrapped around these histones, but it's just getting further condensed and like just packed tightly, right, into like the case that the toilet paper ships in or whatever, right? So this is what's happening to your DNA as it's condensing. This is how it doesn't get all tangled and messed up inside of the cells. So we've talked about this structure. You know, it's this, the A's pairs with T's, the G's pairs with C's, right? And it's in this double helix. At the end of the paper that Watson and Crick wrote, this groundbreaking paper on the structure of DNA and their discovery, they state it has not escaped our notice that the specific pairing we've postulated, and so their postulate, their hypothesis, was that A pairs with T and G pairs with C always, right, and to form these two chains, that this specific pairing immediately suggests a possible copying mechanism for genetic material. What does that mean? And why is that important? I want everyone to think of that, okay? So think about that, and that is going to be the end of this lecture. So go now into the discussion and answer that question.